Welcome back to Biomindscape. Today we are going to know about biological classification. Now what is biology? That deals with the study of living organism. Who is the father of biology? Aristotle. The term biology was first coined by Lamarck and Trivinaris in 1802. And the father of botany is Theopteris and father of zoology is Aristotle. The classification of living organism. What is it? Classification of living organism. Classification is an arrangement of living organism according to their common character. They are placed in a group within a taxonomic hierarchy. And the branch of science that deal with all this is known as taxonomy. Like description, nomenclature, identification, classification of an organism is a taxonomy. It can be numerical, cytotaxonomy and chemotaxonomy. These are the major branches of taxonomy. The numerical taxonomy is based on the characters like number, code. They are assigned and the data is processed by using a computer. Cytotaxonomy, it is a cytological information that is used to categorize an organism. And chemotaxonomy, chemo means here chemical constituent that are taken on the basis of classification of organism. On the basis of reference criteria, the classification of living organism can be of artificial, natural and phylogenetic. Artificial is also known as prior, natural is known as phenetic and cladistic is known as phylogenetic classification. Classification of living organism can be artificial, natural and phylogenetic. Artificial means in this type of system of classification, if there is one or very few character, they are considered as a key feature for classification. Means that these characters, they are taken as consideration for classifying an organism. It never throws lights on affinities or relationship between the organism. That means it is an artificial one. Whereas natural, it is phenetic classification. And phylogenetic classification is also known as cladistic classification. Now phylogenetic classification, it can be of one ancestor, of two ancestors, and many more. So if there is only one ancestor, then it is monophyletic. If there are more than one ancestor, two ancestors, then it is polyphyletic. And the organism which does not include any descendant of the common ancestor, means they don't have that common ancestor and they don't share any common ancestor, they are paraphyletic. Then what is cladistic? It is a method of classifying an organism which is based upon the genetic and the relationship of the ancestor with them. So which are more class significant and they are natural. So the most accepted 5 kingdom system of classification of living organism. What are the 5 kingdoms? Monera, Protista, Fungi, Animalia and Plantae, and it was proposed by R.H. Whittaker. Now, what was national classification? The classification system in which the organism, they are classified on the basis of the vegetative character. They are the group of heterozygous groups that are unrelated organism is avoided. Classification system. Two kingdom system was given by Carlos Linnaeus of animal and plant. What are the merits and demerits of the two kingdom system? The photosynthetic organism, they were included in plant kingdom, and non-photosynthetic organism were included in animal kingdom. What is the demerit of this two kingdom system? That some organisms do not fall naturally either into the plant and animal kingdom or share a character of both. So the three kingdom system was given by Ernst Haeckel, Protesta, Animalia and Plantae. These are the three kingdoms. What are the merit? It created a three kingdom which include unicellular eukaryotic microorganism and some multicellular organism. What is the demerit? Monera were not placed correctly. So this is the demerit. Four kingdom system was given by Copeland, where Monera, Protesta, Animalia and Plantae were given. And what are the merits? The Monera were placed separately along with the other kingdom. And demerits is that the Monera was not subdivided into Archaebacteria and Eubacteria, whereas the sixth kingdom system was given by Carl Woese. There were Archaebacteria, Eubacteria, Protesta, fungi, Animalia and Plantae. What are the merits? The Archaebacteria and Eubacteria were separately placed. Now let's know about the Kingdom Monera. So Kingdom Monera, it is included in all prokaryotic such as bacteria, archaebacteria, mycoplasm, actinomyces, cyanobacteria and rickettsia. So bacteria here, so the bacteria, they are divided into archaebacteria and eubacteria. You know what is a eubacteria? It is a true bacteria, and archaebacteria is a primitive bacteria. The archaebacteria is further divided into methanogenesis, halophils and thermoacidophils. Now methanogenesis is where methane production, it is methane producing bacteria. Halophytes are salty or marine bacteria, and thermoacidophils are present in acid sulfur spring. And eubacteria on the basis of staining behavior, on the basis of structure and on the basis of nutrition is divided. So the true bacteria can be divided into the gram positive and gram negative on the basis of the staining behavior, whereas on the basis of structure it can be divided into the round capsule, spiral and comma like. It is also known as, the rounded one is known as cocci, which is a non-motile one, and bacillus, spirillum and vibrio is of various shape like capsule, spiral and comma like respectively, and they are all motile. Whereas the true bacteria on the basis of nutrition can be divided into autotrophic and heterotrophic. Now here the autotrophic is photosynthetic bacteria, for example the purple surfel bacteria, and heterotrophic is saprophytic, symbiotic and parasitic. The next is, these are archaebacteria. These are the most primitive prokaryote. It have cell wall that is made up of protein and non-cellulose polysaccharide. There is a presence of 16S rRNA, and it makes them unique because it is placed in a separate domain called archaea, which is between the bacteria and eukarya. So archaebacteria can live under extreme conditions like salt pans, salt marshes and hot sulfur spring. So they are also known as the living fossil because they represent the earlier form of life on earth. So it can be used for experimental, for absorption of the solar radiation, for gobar gas production from the dung or the sewage, and for also for the fermentation of cellulose in ruminants. Then eubacteria, also known as the true bacteria, and it does not have nucleus. It lacks nucleus and membrane bounded organelle like mitochondria and chloroplast. So they are usually divided into five phylums like the gram positive cyanobacteria, proteobacteria and Cyprochitis and Chlamydes. These are the five phylum. So the structural detail of typical eubacteria cell is given here. It have a capsule, cytoplasm, ribosome, cell wall, nuclear area, plasmid, flagellum, fimbria, mesosomes, plasma membrane. What it is made up of? The cell wall. It is rigid. Why it is rigid? Because it contains the magnesium ion that bind to the thiochloric acid, and this binding protect the bacteria from thermal injury. The nucleoid, that is the nuclear area, it is amorphous because the mass of the fibrinochromatid type material that occupy 10 to 20% of area of cell. There is a plasmid, it is small circular self-replicating extra chromosomal DNA, have few genes. Then the ribosome is 70S type of ribosome. It consists of rRNA and protein. The cytoplasm, it has 80% of water, protein, carbohydrate, lipid and organic ion. The outermost capsule, it is gelatinous polysaccharide and polypeptide. It protects the bacteria from pathogen and it help in adherence to any surface. Pathogens and desiccations here. It is protecting. The work of the capsule is to protect. Then inclusion, these are the reserved food deposit found in the prokaryotic and eukaryotic cell. These may be glycogen, starch, lipid and sulfur granule. What is the function of plasma membrane? The plasma membrane and its structure and function are similar to that of the eukaryotic plasma membrane, and it is also the site of some respiratory enzyme. What is a mesosome here? Complex localized enfolding of membrane which serve as a respiratory organ, that is the center of respiration. What is this fimbriae? These are short filamentous structure that is composed of the protein pilin, and these are eventually distributed and used for attachment rather than motility. Then flagellum are long filamentous appendages consisting of filament hook basal body, and it is rotatory in function and contain the flagellar protein. So first is respiration in bacteria. The respiration occur in the plasma membrane of bacteria. Here the glucose is breaking down into the carbon dioxide and water using the oxygen in aerobic cellular respiration and other molecules such as the nitrate NO3, and in the anaerobic cellular respiration. Reproduction of bacteria is asexual and sexual. So asexual is by fission, budding and spore formation, whereas sexual method is transformation, conjugation and transduction. So the fusion, what is fission? Bacteria divide both laterally and longitudinally, that is a fission. Budding is what? Vegetative outgrowth that results into the new organism after maturity, that is budding. Spore formation is non-motile spore like conidio, oidea and endospore, they are formed. Sexual method are, although sexes are not differentiated into bacteria, following method are genetic recombination. They are categorized under sexual reproduction in bacteria. Transformation was given by Griffith in 1928, where the genetic material of one bacteria is transferred to other through conjugation tube. Then conjugation, it was given by Lindeberg and Tatum in 1946, where the transformation of genetic material occurred through the sex pili, and transduction was given by Zinder and Lindeberg in 1952, where the transformation of genetic material occurred by the bacteriophage. What is economic importance of bacteria? Some bacteria are useful in producing various useful substances like curds, cheese, antibiotics and vinegar, etc. So while other bacteria cause several chronic diseases in human, plants and other animals. Now we are going to see the structure of mycoplasm. It was discovered by Knockard and Rocks in 1898, and these are the cell wall, less aerobic and non-motile organism. They don't have a cell wall and pleomorphic nature. They are known as the jokers of living world. Why? Because mycoplasm are known as the pleuropneumonia like organism, that is PPLO. So these are the small living cell yet discovered can survive without oxygen and are typically about 0.1 mu meter in diameter. Now we can see the structure here. There is a lipoprotein membrane that is of 3 layers. There is a ribosome, the DNA and a soluble RNA is present in the structure of mycoplasm. One of the most important structure of cyanobacteria blue-green algae is they are gram-negative photosynthetic prokaryotic which perform oxygenic photosynthesis. They can live both in the freshwater and marine habitat, and they are responsible for the bloom in the polluted water, that is eutrophication. They have a photosynthetic pigment like chlorophyll A, carotenoid, and phycobilin, and food that is stored in the form of cyanopaquin starch, lipid globulin, and protein granule. These cyanobacteria have cell wall that is form of the peptidoglycan, naked DNA, 70S ribosome, and the absence of the membrane-bound organelle like the ER, mitochondria, and Golgi. So the Red Sea is named after the coloration provided by red color cyanobacteria. Can you name that? Trichodesmium erythrium. This is the name of this particular cyanobacteria which give the coloration of the Red Sea. Then cyanobacteria can fix atmospheric nitrogen through a specific structure. We call it as heterocyst. So these are modified cell in which the photosystem 2 is absent. Hence no cyclic photophosphorylation. This does not takes place. Non-cyclic phosphorylation does not takes place here. The nitrogen fixation is performed through an enzyme. Nitrogen is present in it. Rickettsia, these are small aerobic and gram-negative bacteria. They belong to the phylum Protobacteria, which are capable of growing in lower level of nutrition and have a long generation time that is relative to gram-negative bacteria. Rocky Mountain Spotted Fever, RMSF. It is a tick-borne human disease caused by Rickettsia rickettsii, an obligate intracellular bacteria. Thanks for watching. Do like, share and subscribe BioMindscape.